Thank you guys for joining us today. We have an intimate group going to be joining us for our work in progress. So the goal of these webinars is to provide early career researchers with an opportunity to present their work and get some feedback from our community. And so we have two really wonderful speakers lined up today from the University of Colorado. And so I'll introduce each one and then we'll jump into their talks in just a minute. And then moderating today is Dr. Julia Woodcock from NJ Health, who is going to be helping facilitate discussion. So thank you, Dr. Woodcock, for joining us. So our two speakers today will be Dr. Christine Farrell, who's a physician scientist postdoc at the University of Colorado. And then after her talk, we'll have Dr. McKenna Burns from the Goose Denmark lab, who will be speaking as well. So why don't we start up with Dr. Farrell? Go ahead.
Yeah. Well, thank you guys for attending. I'm interested to get your thoughts on a project I've been working on for a little bit, and thanks to the moderators for inviting me. I will be talking about my work, specifically looking at the complement receptor C3AR and then its importance for hypoxia-induced pulmonary hypertension. I'm one of the physician scientist postdocs at the University of Colorado. And so the CBP at the University of Colorado has really been interested in inflammation as a driver of pulmonary hypertension with this kind of commonly cited paper from the group showing that the degree of vascular inflammation around the pulmonary vessels of patients with pulmonary arterial hypertension correlates not only with kind of the degree of vascular remodeling, but also correlates with the degree of the mean pulmonary artery pressure. What's a little bit less cited from this paper is that 50% of all vessels analyzed had these small microthrombi within them. And so our group has particularly been interested in looking at vascular thromboinflammation with platelets as being the bridge between thrombosis and inflammation as a potential driver of pulmonary hypertension. And so just a brief background on basic platelet biology so we're all on the same page. Resting platelets kind of float through our vasculature in this resting state. When they encounter damaged endothelium, they then assume this activated state where they change morphology into this spindle shape-like morphology. And they then release a variety of cytokines and kind of change the activation status of cell surface receptors to help not only kind of do linking to other platelets through this alpha 2b beta 3 linker through fibrinogen. And they also release both alpha and dense granules that are kind of chock full of a variety of cytokines in addition to complement. And we'll be looking not only at complement, but also this cell surface receptor p-selectin, which binds to various leukocytes. And so when platelets become activated, they can bind to other platelets to form these platelet plugs, which what we usually think about when we think about platelet activation. But they also release extracellular vesicles full of cytokines and then can bind to neutrophils and monocytes themselves to help both recruit these immune cells to the site of vascular injury and then also activate these immune cells. And in particular, there's a variety of receptors that can do this, but we'll be talking about p-selectin, as I talked about previously, which binds to PSGL1 on these leukocytes to help recruit and activate them. And so this came out last year showing that even despite our kind of modern therapies, thrombocytopenia is a marker of worse survival in patients with PAH. Those that have lower platelet function have worse survival kind of across a few years compared to those that have normal platelet counts. And the thought behind why this might be is this group looked at patients with decompensated RV failure and found that their platelet counts in their blood decrease during episodes of decompensation, but then their platelet numbers within their lungs increase. And so a lot of these platelets are trafficking to the lungs at times of decompensation as measured by kind of platelet staining in brown here. And then also looking at electron microscopy, they're kind of filling the pulmonary capillaries. And so our kind of thought was, what are these platelets doing during these times of decompensation, and can they help be driving inflammation and worsening pH? And so our group looked at if you deplete platelets in a murine hypoxia model of pH, can they have prevented kind of evidence of RVSP and Fulton's index? And we did find that giving these mice an antibody which completely eradicates their platelets for at least 72 hours. If we place them into hypoxia, the usual increase in RVSP at day 21 is attenuated in these platelet depletion mice, and they also have attenuation in the usual increase in Fulton's index at day 21. Interestingly, if we looked at interstitial macrophage accumulation within the lungs of these mice, they also had decreased IMs in their lungs compared to wild type controls at day 3, indicating that platelets may be kind of helping to perpetuate the inflammatory signal early in inflammation. And so the first thing I did when we looked at the group was kind of looking, is this relevant in humans? And so we did lung stereology to look at where are platelets within the lungs of these remodeled vessels of patients with kind of end stage PAH. And we found that there's increased platelet density as measured by CD41 staining in yellow in the perivascular space of patients with PAH compared with controls. And this was excluding kind of just like intraluminal thrombosis, but looking in the perivascular space itself. And then when we look at these lung platelets, they're often co-localized with leukocytes, which is measured in CD45 in red here, and then particularly macrophages in CD68 in white, kind of looking at this pink color, that there's co-localization of kind of both yellow and pink, showing that they tend to be co-localizing with them, and we assume kind of binding to and interacting with these leukocytes. And as a potential kind of jump starter of this, we've been interested in complement as a marker of kind of platelet activation. The Stenmark group has previously found that there's increased complement deposition in the lungs of patients with PH compared with controls. And then looking a little bit more basic science, if you take mice that are lacking the complement protein C3 and expose them to hypoxia, the usual increase from normoxia to hypoxia in black here is attenuated in these mice lacking C3. They also have attenuated increase in Fulton's index. And then very interestingly, there's usually platelet activation in these mice exposed to hypoxia, as shown by platelet cell surface expression of P-selectin. And there's absolutely no platelet activation in these C3 knockout mice. And so my question when I joined the lab is kind of what is mediating this? Can we target this a little bit better, since C3 is a linchpin and knocking it out completely can have some kind of serious consequences on the immune system. So just looking at the complement cascade, so we're all on the same page, there's kind of 3 main inputs to complement activation. They all converge on this molecule C3 that I just mentioned. C3 is then cleaved into 2 particles, C3a and C3b. C3b goes down and kind of perpetuates the complement cascade to form these small pores called a membrane attack complex to lyse cells. And then it also kind of spits out this molecule C3a. This binds to the C3a receptor known as C3AR, and this is present on leukocytes and also platelets, and has been thought to kind of help recruit inflammatory cells to the site of injury to help recruit them and activate them. And so we were wondering, if you target C3a, which is a little bit more specific, is that mediating this effect previously shown on C3 with platelet activation and kind of protection against pulmonary hypertension? The Stenmark group has previously found that the C3a receptor is highly expressed in the perivascular space of rats exposed to hypoxia, both in terms of staining of cell surface receptors and in terms of mRNA expression. However, it was not really known what cell types were expressing this receptor. And so when we looked at single cell sequencing data of mice, just kind of normoxic mice, We found that macrophages are the number one expressor of this receptor with IMs up top, AMs down here. And then kind of the second most common cell type expressing it are platelets. And so we were really interested in looking at both of these cell types expressing this receptor to see kind of what's mediating this. And so my hypothesis was that hypoxia-induced platelet C3a receptor activation results in lung platelet accumulation and then platelet macrophage aggregate formation, and this drives pulmonary vascular inflammation and remodeling in pulmonary hypertension.